The data that I'm going to talk about today actually sits in a data archive that is managed by the federal government, right? So that, in terms of gatekeepers to access, a term I love, means something a little different when the federal government is the gatekeeper to access in terms of what type of punishment might happen if you misuse the data. So I'm going to talk about those types of issues. I think when I first put this title together, that I was probably thinking that I might talk about the Brain Initiative data. I'm not going to do that at all, but I do co-lead the group of program staff that manage the Human Brain Initiative in the US, and I'll be happy to talk about that and our data activities later if there's any interest. So a couple of guiding ideas that I think I've picked up in a variety of locations, although I'm not sure I have good academic citations for all of these, but they do certainly guide the way that I think about sharing data. Number one, research subjects I think really want their data to be shared broadly. And I think they're not too concerned about how researchers use their data, although as we've talked about, they may be very concerned about how people who are perhaps less trusted than researchers use their data. Second idea is that the diseases we're trying to understand today are complex, and by that I mean just because you have a symptom, there can be a variety of underlying biological causes that give rise to that symptom. And so, to the extent that that's true, you really need to have lots of data from a variety of different sources, because finding the subpopulations within a group that shares a symptom where you don't understand the underlying biology is impossible unless you have data from many individuals. Number three is that differences in data sharing laws can make it difficult or impossible to move data across international borders. There's a simple solution to that, and that is federating data archives that sit in different locations so that the data doesn't move but can be queried. Simple in some ways, technically not necessarily so easy. Finally, I think there's a real urgent need to understand complex diseases from the large data. Second couple points, having run a large repository for a while, it's pretty clear that policy considerations can be used by an excuse not to share data. And something that I think I disagree with a bit, I think most researchers, neuroscience researchers, still really don't want to share their data. I think that's more than half, at least. I think it's changing. It's certainly gotten better, but this is not a problem we've solved. So what I'm going to try to do today is talk briefly about two different data archives that have built infrastructure that's needed to share data on complex diseases. One is the NIMH Data Archive. The second is the Human Connectome Program, and I'm going to certainly try to leave enough time for good discussion. So the NIMH Data Archive stores data from experiments involving human subjects, as does the Human Connectome Project. That's the reason I'm not talking about the Brain Initiative. The early awards that we've made for the Brain Initiative don't involve human subjects, and so I thought it was better to talk about these two. And it is, as I said earlier, a federal data repository, meaning that the federal government owns the data. Most subjects that we have data from have consented to broad data sharing, and I think that gets back to some of the points that Alan was making, that when you have a situation like that, your life is easier, although our life is going to get more complicated because we're about to take in data that doesn't have these sorts of consents. And the archive is set up so that it can really store any sort of data from human subjects, and I'll show you how that looks like. That's what the website looks like. We have a couple of different entry points that are kind of set up with queries to try to facilitate. If you're interested in clinical trials, you would go in through NDCT. We think that, but all of no matter where you start, you're querying against all of the data in the archive. All right. So at the moment, we currently make data available from over 200,000 subjects, and you might think, well, God, he's already got 200,000 subjects. That should be enough to understand any complex disease, and my sense is that that's probably an order of magnitude less than we really need. We are storing about a petabyte of imaging and genomics data in the Amazon cloud. It's the federal portion of the Amazon cloud, which has slightly different security aspects than the normal portion of the Amazon cloud. And we don't have any personally identifiable information at the moment, although I expect that to change. And the way we think about the data archive, even though this is absolutely not the way that it's set up in the underlying infrastructure, but it's easy to think about this as a two-dimensional matrix where the rows are an individual subject, and they have what we call the GUID, the Global Unique Identifier, and I'll talk about that. And the columns are individual questions from data dictionaries, clinical assessments, or it could be links out to imaging data, or it could be raw data from an EEG experiment, or it could be derived data from any of those sorts of experiments, right? So that's a good framework. You start to see part of the problem, and I'll talk about this now. We have on the order of 180,000 data elements. That is, we have 180,000 columns, right? And we have 200,000 subjects, right? So you wouldn't know this unless you had tried to aggregate all of the data in a particular domain. But it means that we collect, as researchers, data in ways that it just can't be justified. There's no reason to have the number of data dictionaries that we have in mental illness. Our science isn't precise enough to need over 2,000 data dictionaries. The world, this is a personal opinion now, the world would be a much better place if we had fewer data dictionaries and we just iterated on one data dictionary to make it better. All right, so the data dictionaries are the columns. As I said, we have over 2,000 of them. They describe anything that a researcher wants to do. And we don't, at the moment, have any limitations on the creation of additional data dictionaries. We curate them, and by curation I mean, I'll show you a good example in a minute. If you want to see all of the subjects who exhibit excessive repetitive motion, we do that type of curation. It's difficult. It takes a lot of time. A very significant fraction of my staff time goes to data curation. We have a validation tool, which is, I think, a very useful tool. And I'm going to talk about rigor and reproducibility a little later, but the validation tool really helps labs make sure that their data conform to the data dictionary that they think they're using. All right, so here's what the data dictionaries look like a little bit. You see some of the different binnings of different types of data dictionaries. These are individual data dictionaries. These are all freely available, so if you go to the NDA website, you can click on any of those. When you click on one data dictionary, you get all of the questions in that data dictionary. You get the allowable range of values for those questions. And that's what the validation tool actually looks at. In the case of the one that's being pointed to, the allowable values are between 0 and 2. And if someone tries to run a data set through the validation tool and there's a 3 in that column, or an A, or free text, the validation tool says, no, that's not right, and you need to, then immediately that, that there's a problem. We also make, getting back to who can see what types of data, and I'll be a little more explicit about that, but we make summary level data available to anyone with a browser. So this is the range of values for one question in one of those data dictionaries. And the nice thing is that when you have 200,000 subjects in the database, the bars in each of your, for many of the data dictionaries, the numbers of subjects that have any particular answer to any particular question are in the hundreds, and that's enough data that would be very painful in general to collect yourself. All right, the GUID, the Global Unique Identifier, is the other building block, and this is a piece of software that sits at the research site. What it does is it takes some information, personally identifiable information, first name, last name, date of birth, place of birth, sex, and it creates a one-way hash of that data. One-way meaning that you can't take the hashed data and reconstruct Greg Farber, born in Harrisburg, Pennsylvania, but we get the hash code is then sent to us, and we simply maintain a list in a very secure location of hash codes and random identifiers that we've associated with that hashed code. And so all we do is, if we get a new hash code in from some lab, they get a new random identifier. If we've seen that hash code before, they get the same random identifier that we have sent out the first time. And what that lets us do is aggregate data from subjects who are seen in multiple different laboratories. And this just shows you that it works. This is actually an example of two things simultaneously. This is data that's in the Interactive Autism Network database. We don't actually hold this data. So this is a query using our deep federation tools into Ian. And this just shows the Ian is a database that has mostly clinical phenotypic data reported principally by parents of autistic children. The parents aren't uploading neuroimaging data, and they're not uploading genomics. So what this shows you is that some of those subjects have been seen in research studies, and those research studies have done genomics experiments or have done imaging experiments, right? All right, so at this point in NDA, the data's been received. We've got GUIDs, hopefully. If we don't have enough information to actually make a GUID, we assign a pseudo GUID. So what do queries look like? Well, we allow queries in a couple of different ways. One is all of the data from a particular laboratory. So this is what a laboratory's worth bucket of data looks like. We call it a collection, and all of those links that I've highlighted link out to other databases that have useful information. The first to the NIH grant number. Not all of our data is paid for by NIH. We hold data from anyone who can deposit it that has relevance to mental health. It goes out to the publications associated with that grant, so if you click on any of those, you go to PubMed, and so you can read the journal, and it goes to the data dictionaries. Data from papers is a second way that we allow queries. And this is what a data from paper looks like. So this is the data associated with a publication. And as I think I said earlier at the question time, we assign DOIs to each of these data sets, to these studies. And the DOI, you can click using some tools made available by DataCite. You can count clicks. It's not everything you need to get this to be as good as a publication in terms of getting credit for sharing your data. But I don't think there's too much additional infrastructure that's really needed. And while I don't believe that giving credit for sharing data is going to solve all of our problems, it would be a good step, a good next step for us to take. It's also, I have to say, it can be very interesting to look at the data that's in a collection from a laboratory and compare it to the data that's in a study. You can start to then ask questions about, or you can start to get some sense about subjects that were measured but not used and things like that. And finally, I should say that the data in a study doesn't all have to come from a single collection. Different labs that have deposited relevant data will show up in a study, and they will get credit. Each of the data measures will get credit. And finally, I'm going to talk briefly about this query by concept. So this is exactly the reason why we don't need to generate any more data dictionaries, at least not in mental illness. This does show a variety of questions that all have the phenotype excessive repetitive action. And so you see that there's a data dictionary question number and the allowable range between 1 and 2. So a 1 and 2 in that data dictionary is the same as a 1 and 3 for any of these questions from a different data dictionary, which is the same as moderately, quite a bit, extremely in that data dictionary, which is the same as 1 to 3 in that final data dictionary. You can imagine how painful this is. Maybe you can't imagine how painful this is, but this is really tough. All right, talks about some issues related to policy, mostly in the NIMH Data Archive, but also a little bit in the Human Connectome project that I'm going to get to. So my assertion, and again, this is not a government official speaking, this is my assertion, that consent language that restricts the use of data for a particular purpose results in profound negative consequences. We see examples like this all the time, where someone who, if you have a data set that's consented for autism use only, and a researcher comes in and says, I want to study the group of subjects that have common symptoms and either have a schizophrenia or autism diagnosis because I'm interested in something or other, they probably can't access the autism data set. Restricted data certainly puts a lot of data off limits for data mining types of experiments, and you don't then know what the consequences are for not having that data in your study. I think there are a few cases where you can make a legitimate case that a restrictive consent might be appropriate, but in general, these should be rare, and in the moment, in science, in neuroscience, they're not rare. We talked a little bit about depositing data, I think, earlier, and the third point maybe is the most important one. Asking people to volunteer their data probably just isn't good enough, because it does take time and effort to put your data somewhere, and researchers need to get credit, because it does take effort. Being a funder helps, because we can make it a condition of getting the grant award that you will in fact make the data available. All right, so how do you actually submit data, and how do you get access to data in the archive? And this is something that we've struggled with, although the news might be a little better. I was told early on that if you put any bureaucratic barrier between a researcher and getting access to the data, they will not ask to get access for the data. They'll just go on and do something else, right? I don't think that's actually true, and I'll show you one data point that suggests that isn't true. But for both access and submission, the PI has to sign a form that says that they're going to treat the data with respect, and I can define that a little better, but it means no re-identification, no redistribution, the sorts of things that you might expect. But the institutional official also has to sign that form, and that means that the research institution itself is on the hook to make sure that the data are treated with respect. When you're a government funder and a research institution has made a promise to you, in cases of violation of those promises, there are real consequences. And that it can be true at times that the government is a very good access gatekeeper, I think. All right, data from institutions outside the US, we do have a little bit of it. The reason we have it is that an institutional official said that giving you this data is consistent with the way that the data are collected and with the locally applicable laws. Timeline for sharing data. I think the important thing here is that you can't wait until the end of your experiment to submit the data to a data archive. Two things happen when you do that. A, you run out of money, and so you've spent it on doing research, and so you don't necessarily have the funds that are needed. And secondly, you haven't validated your data against the validation tool, and so you haven't found the errors early on, and that's a big negative. All right, so I'm going to very quickly, because I want to make sure we leave enough time for questions. Jan brought up earlier that there's a rigor and reproducibility crisis in science, and NIH has recognized this. I think data archives help in a couple of ways. The first, we've talked about, the validation tool really helps labs discover when they have problems with the data that they've measured. The second is it's very interesting, as I said, to look at the data that are related to a publication. If you don't think that paper is right and there's a study that's been created for it, you can look at the exact data that were used in that paper the day that paper is published, if you have access to the database, and you can run your own processing pipelines and do whatever you think is needed to either prove to yourself that you're wrong or to prove that the data should be interpreted in a different way. You know, something that I've discovered fairly recently is that the GUIDs also have a role in this rigor and reproducibility. This is shocking, but true. There are professional research subjects who make a living, not necessarily, this is in the US, not necessarily a good living, but a living being human guinea pigs. They lie about things like, do you hear voices? That's a great symptom to be able to admit to. One or two of these people in a study of 50 or 100 subjects invalidates the study, basically. So the GUID helps find these people, and there are other commercial services that also help with this, but I think this is an under-recognized issue. The second issue that's not on this slide is that when you see a paper about autism, you tend to think that the subjects in that paper have been selected at random from a large sample of all individuals with autism. But we've shown that there are a number of labs that are really good at recruiting autistic subjects, for example, and they make those subjects available to others in the field. So papers that appear to be uncorrelated may actually be much more correlated than you expect. The GUID unveils this sort of problem. All right, here's the staff that, my enormous staff in Bethesda that I just get to come to nice places and give talks. They do all the hard work. Right? The Human Connectome program, I want to compare and contrast a little bit with what we've done in the NIMH Data Archive. So this was a large research project paid for by NIH to collect data in a standardized way. And because we're a little short on time, they did really great work in creating the next generation of MRI imagers. In fact, the Prisma that is being sold by Siemens is a direct result of the work that was done in the Human Connectome project. But their real goal, after they got to play around with building a new device, two new devices, both of which are spectacular, was to measure data from a group of 1,200 subjects. And NIH was so impressed with this that this first group of data from 22 to 34-year-olds that we've expanded it now to cover the whole lifespan with a series of additional awards, as well as a series of awards to measure the same connectome, use the same data collection methodology in subjects that have a diagnosis of some sort. So we're soon going to have 2021 is really pretty soon. We're going to have something in the neighborhood of 8,000 subjects and 12,000 scans, all of which are collected pretty much the same way. The Adolescent Brain Cognitive Development, ABCD study, which is another enormous NIH project, voluntarily chose to use the human connectome data collection protocols in their study. We've just released the first data from the ABCD study, and that first data set has 5,000 kids. The full data set will have 10,000 kids with longitudinal measurements. So this data collection methodology is becoming something of a standard. It won't last forever, but it's pretty good for right now. So the data is in the Connectome Coordination Facility at WashU, run by David Van Essen. And they do similar things. They've got an intake process, and at the moment they're making data available, but that's going to change, and that data is going to be moving into our archive. What I wanted to show these last couple of slides mostly for was to show you that a lot of people have used this data set. This is terabytes downloaded, right? So that's a petabyte of data. They were doing data downloads with disks, actually shipping disks, and they shipped a lot of disks, right? Something that is important for the HCP was that in order to get access to this data set, you just need to self-certify that you're a good person and you're not going to do anything bad with the data. You don't have to be at a research institution. Anyone with an email address can self-certify. The good news is that meant that data got around the world, right? I mean, people really, literally, each of those dots is someone who has used the data set, has access and is using the data. The bad news is that it's hard to impose penalties. The only penalty they have for treating the data badly is to say you can't access the data anymore, but if you've already got your connectome in a box on your shelf, that requires that someone go and launch a complaint with an institutional official, ... right? I think the key question here is, in thinking about open access and more restrictive access, is what penalties need to be imposed if the data isn't used appropriately. I think that's an important point to think about. That's the consortium team. The first round was led by David Van Essen and Camille Ugerville. And it really was a great group. Deanna Barch played a key role in the data collection, the data dictionaries, and in other ways. So in summary, and I think I've left enough time for questions, you need a lot of data to understand complex diseases. And I really think you need data from millions, not from hundreds of thousands. Informed consents, national laws concerning data sharing, and investigator preferences can all restrict aggregation of data. There can be some technical solutions to some of those problems, but it requires effort. If you have an option about sharing data under a very open access model or in a federal database, I think you can make that decision based on what you or the research subjects would expect to happen based on if the data is misused in some way. And finally, something that was quite a surprise to me, even though it's much easier to get data when you just need to give a repository a website and say, I'm a good person, for ABCD, that's not true. You have to go through a data access process. And in looking at, now the ABCD data has only been available for about 4 weeks. So this is one data set at a very early time point. But at the moment, it looks like we have as many people who have access to ABCD data as the Human Connectome Project had at that point in their life cycle. And so I think that it's not necessarily true that open access is the only way that you will get researchers to use a data set. If the data set's of high enough quality, I think that they will go through the bureaucratic hurdles that are needed to get access to the data. And that is my last slide, so thanks.